Hi there, my name is Miles Turk, and today I'll be telling you about the experiments that I performed with a member of other chemists, which was the synthesis of isopentyl acetate, also commonly known as banana oil. So let's get started. Esters are covalently synthesized from carboxylic acids, an alcohol, and the presence of an acid catalyst. And when performing a procedure, it is extremely important that you do all the steps in order and do not miss any, because missing a step could alter your results in the end. Isopentyl acetate is synthesized with an equilibrium reaction, and this reaction is between acetic acid, isopentyl alcohol, and a catalyst. And we sped up the synthesized reaction with the catalyst and heat from our microwave reactor. And then once we ran our sample through the microwave reactor, we ran it through the HNMR to confirm that isopentyl acetate was indeed produced. Alright, so what a microwave reactor does is it allows you to heat your sample from the core and out, which helps save time and energy. And how it does this is you place your sample in the microwave reactor, which is hooked up to a computer. With this computer, you can set a specific temperature and time period that you want your sample heated to and for how long. And then once it's heated for that long at that temperature, you can set a specific temperature that you would like it cooled down to with an air compressor. And how HNMR will help us determine that an isopentyl acetate was indeed produced is that HNMR data allows us to see the different types of hydrogens on a molecule. And since we know the structure of isopentyl acetate and all the different hydrogens on that molecule, looking at the data, we can confirm that indeed our isopentyl acetate was produced. And how this works is protons are lined up with a magnet, and then they're hit with radio waves, and these radio waves knock them out of their alignment, and as they are moving back into alignment, the HNMR picks up the frequency at which they move, and all these different types of hydrogens come back in alignment at different frequencies. So what HNMR tells us is, first, the different types of hydrogens, which is how many different peaks are in our graph. Second, what those hydrogens are next to as an electronegative elements, and that just tells you how far downfield your hydrogens are, which means they're next to an electronegative element as in oxygen, nitrogen, fluorine, things like that. And the closer they are to those, the farther they move downfield. Third thing that it tells us is the splitting of these hydrogens, which means what hydrogens are next to each other. How close they are. They usually are 3 bonds away and they can interact with each other. Not so much as they're hitting each other, but they contort how they align. And the fourth thing to tell is the integration of the hydrogens, which is just how many different hydrogens are in each peak that you see. So what we did was we took 4 milliliters of acetic acid, 2.8 milliliters of isopentyl alcohol, 20 drops of sulfuric acid, which was our catalyst, and 2 grams of silica beads, which in the end was used to soak up the remaining water. Because when this reaction happens, our two kind of products that are produced is isopentyl acetate and water. And the silica beads will help to soak those up, so our remaining solution will hopefully just be isopentyl acetate. And so we placed our sample in a microwave reactor and we set the computer to 130 degrees Celsius for 15 minutes. After those 15 minutes had gone by, our sample was then cooled to 50 degrees Celsius, and after it had been cooled, it was ready to run through the HNMR. So the data we found was our graph showed that something went wrong in our procedure because the amount of peaks, how they were integrated, was off. You could tell that by looking at it right away. So we had to analyze it a little more. So with our first peak, we saw that there were 6 hydrogens. That was the integration of 6 hydrogens at a doublet, which means that it was talking to one other hydrogen. And our second peak showed that it was 3 hydrogens, and it was a multiplet, which means that there was 1 hydrogen and 2 other hydrogens that were so similar. They weren't on the same carbon, but they were so similar that they acted the same way. our third peak, which was one of the first things I saw when we looked at the graph and helped me to determine that something went wrong, was integrated for 6 hydrogens at a singlet. And the only singlet that we had on our molecule should have only been integrated for 3 hydrogens. So the fourth thing we found was that the fourth peak that we had was integrated for 2 hydrogens, and this was a triplet, which makes sense because we had a carbon with 2 hydrogens that were 3 bonds away from 2 other hydrogens, making it triplets. That makes sense. And the fifth peak, which really sealed the deal that something went wrong and something was left over in our sample, was 1 hydrogen that was a singlet, and it was super downfield at about 8 ppm. And what this told me was that there was an OH group in our molecule, and our product should not have had an OH, which led me to believe, and some of my partners in this experiment, that there was leftover acetic acid in our solution. And so what went wrong was, when we were doing our procedure, we missed the step and when we should have washed our sample after it came out of the microwave reactor in 10% sodium bicarbonate, which would have neutralized the acetic acid, making it essentially water and then soaked up by the silica beads. So I found that we did indeed produce the isopentyl acetate, but we skipped a step of the procedure in which we would have removed the remaining acetic acid, which led to our data. So missing a step in a procedure can alter your results, but these results that you get are not useless. It is important to determine what went wrong during your procedure in order to not make the same mistake in the future and to help you become a better chemist. Any questions?